Hi, everyone. I'm Brian Gratwicke. I am the amphibian conservation biologist here at the Smithsonian National Zoo and Conservation Biology Institute. I first arrived at the zoo on a cold winter's day in 2004, and I was rescued by the kind folks in Amazonia who said, "Here's a nice warm place where you can look at wildlife all day long." So I started out with Ed Smith as a volunteer. And I think that's where I first met Randy. So it's been a long time, but I really wanted to break into conservation. That was my objective for moving to Washington, D.C. So after about 5 years, I eventually did land a job at the National Zoo to lead the amphibian conservation efforts. And this program was set up in 2008, which was Year of the Frog. And AZA led a huge campaign to persuade other zoos to get on board and try and build an amphibian ark for the frogs. So I was hired. I was given 6 months of salary, a desk, and a computer, and told to go save the frogs. And if I was successful, then I could stay. So I'm pleased to report that I am still here. And our most recent project was here in Panama in the Campana National Park. It's Panama's first national park that was created. Beautiful montane habitat. It is also the home of the Panamanian golden frog. But we have done an outrageous thing. We attempted to reintroduce 4 species of frogs at the same time. So the first idea was to see if we could restore a population of Pratt's poison dart frogs that had gone extinct from this national park. They had disappeared. Our project director, Roberto Ibanez, had studied this frog for his PhD, and he had studied the calls of this frog in Campana National Park. And we spent 2 years looking for them and using passive acoustic monitoring and AI to see if we could to relocate this frog, and we couldn't. But we had found a population somewhere else where the frogs had recovered. This is Bajo Bonito, and we got with our partners at the Cheyenne Mountain Zoo and Zoo New England, and we made a little expedition out there, and we managed to collect 12 pairs of animals, male-female pairs, and we moved them to, do we get sound here? But we'll try that again. So we moved them to Campana National Park, and you can hear the sound of Campana National Park restored. And it was a really magical moment. These frogs were kept in these kiddie play tents for 2 weeks, and then they were released. And at the time that we were, here's a frog calling on a rock inside the kiddie play tent. They rather liked their digs. And if you look carefully behind this female frog, she's sitting on a clutch of eggs that was laid inside the kiddie play tent in the 2 weeks that we were just acclimating it to the new environment. And she took the tadpoles onto her back. And when we opened the kiddie play tent, she went off and took her new clutch of eggs into the wild. So that's probably got to be the fastest reproduction following a translocation that we've seen. We also tried a pilot release of crown tree frogs. This species was last seen in the wild 15 years ago in Panama. And so we have these breeding in captivity. They're doing pretty well in captivity. And we have worked with our Reptile Discovery Center, and I recognize Ed Bernikowski here. He's been a great champion, along with Matt Evans, from the Reptile Discovery Center. They have really been the people with the know-how. They know how to care for animals in our collection. And this is it's an art, it's not a science. You have to know and understand your animals and get them to reproduce. And these guys have been magic. This is Matt in one of our amphibian rescue pods in Panama. We have 2,000 adult frogs breeding in our collection in Panama, and most of them have disappeared in the wild. So here we have Nate Weisenbeck. He has been leading the radio tracking of these frogs. What we did is we basically built little artificial tree holes. They like to breed in tree holes, and mom likes to feed unfertilized eggs to her tadpoles. So this animal got habituated to these little artificial tree holes. We built a little mesocosm around the artificial tree holes so she could get acclimated to the new habitat. And then eventually we just opened up the front door of that and let them out, and we radio tracked them. So they have done very well, and using our passive acoustic monitoring, we have been able to restore the sound of this frog to the forest, which is a really incredible moment for us. Our third release trial was lemur leaf frogs. These have been doing pretty well in captivity, beautiful frog, and they have lots of tadpoles. And so what we did is we decided to get these big cattle tubs and stick them in the rainforest and release some tadpoles into the cattle tubs. And we put a little antifungal solution because they disappeared because of the amphibian chytrid fungus. So in half of the cattle tubs, we put some antifungal solution. And the other half, we left them untreated. But what we have seen is 95% survivorship of these tadpoles with and without the antifungal treatment. So they've been doing great. And when these guys come out of the water, they have these little frogs on the side here. And then there's one of our little metamorphs being released into the wild. Very rewarding, this work so far. And the final species is the Panamanian golden frog. This species went extinct in the wild before I started at the National Zoo in 2009. So I've never had the honor of seeing this species in its native habitat. Here's our project director, Roberto Abanez, releasing some golden frogs into their kiddie play tanks. And they have been doing pretty good. And this is a very special moment for me because it's the first time that I got to see a Panamanian golden frog in the place where it's supposed to be, not inside a glass box. So that is just a magical moment for me. And I had this picture in my mind's eye for 15 years, and I finally got to see it last week. So thanks very much to our team who has been working really hard on rearing these animals and preparing them for release and working on all of the different ideas, all the crazy ideas we worked on the last 15 years trying to solve the chytrid problem. We're just trying everything we can with all the resources we have, and we're just hoping that it'll work. So stay tuned.